Okay, so this unit, evolution, is actually a combination of a few chapters in your book. And what I've done is I've gone through and sort of picked out what's most important as far as the mechanisms by which evolution occurs. So we're going to start simple with Charles Darwin. He's considered the father of evolution, and his theory was natural selection, or survival of the fittest. And so he traveled to the Galapagos Islands on a ship called the HMS Beagle, and he was a naturalist. He was basically just looking at all the different organisms and cataloging them. But he noticed that there were fossils for example, tortoises in the rock layers, and he saw that there were changes in the ancestral species of tortoises compared to the ones that were living on the islands. He also observed all these different species of birds. He's famous for his finches that he wrote about. And so he came up with this theory of natural selection, which basically says, first of all, there's genetic variation in members of a population, meaning we don't all look alike. Secondly, he used some evidence from another guy named Thomas Malthus, who discussed populations, and he basically said, Thomas Malthus, that like, for example, if we started with a pair of elephants and every female elephant had the maximum number of offspring that she could have, and they all survived, that there would be too many elephants after, 100 years or something, that there would be so many elephants, there'd be nothing, no room for anything else on the planet. So Darwin agreed that there more offspring are born than actually end up surviving and reproducing. And the reason why this happens, other than, of course, mutations and disease and other things, is that there's competition for basic needs, food, water, shelter. There's not necessarily enough to go around for everybody, and so only some may survive and some don't. So he basically said that those individuals that have better characteristics would survive longer and therefore reproduce more, and that would mean that they would then pass on those good characteristics to the next generation in the population. Which brings me to a really important thing, a misconception, is that number one, that individuals do not evolve, populations evolve. If it suddenly became really, really cold, I'm not going to grow fur and suddenly survive. But these people here that have a mutation, these people I think they call it like Werewolf Syndrome or something, but it's a genetic mutation, and they are covered in hair. Now, when we look at them now, people think of them as freaks, and unfortunately, they have to get jobs with the circus or whatever, but if it got really, really cold, these people would probably survive better, and that would mean that there would be more of them passing on that gene, and so the future generations, maybe humans would look like these people, because those would have been the genes that got passed on. So individuals aren't going to change. I am born with the genes that I have, but because there is variation, there may be other people that have genes that make them more fit for a particular environment than the genes that I have. So that would be what Darwin's talking about, and that is a common misconception. And the other thing that I should say before we move on is that we don't evolve on purpose. There's always going to be variation and random mutations that are 99% of the time either neutral or negative, but maybe that mutation is beneficial, or it's neutral and becomes beneficial in another generation because of a change in the environment. So we don't evolve on purpose. For example, when we look at antibiotic resistance, it's not like you expose the bacteria to the antibiotic and then they develop a resistance to it. There were already some individuals that had a resistance to the antibiotic before they were ever exposed. Those are the ones that survive, they pass that trait on, and now you end up with larger populations that are resistant, but the resistance doesn't come about on purpose because they were exposed to the antibiotic. So that's really important. Okay, so natural selection sometimes can be split into three separate types of natural selection. So the first one is called directional selection, and that's basically where one extreme is favored. A lot of times the example that's used of this is the peppered moth. And there's actually, if you look it up, there's some questions about this whole peppered moth experiment and how there were problems with it, but books still use this as the example. The idea is there were always light and dark moths, but the light moths blended more with the trees because the trees had these things called lichens growing on them, which is sort of like this algae fungal symbiotic growth. And you can see how, in this case, the lighter moths would blend better. But when the Industrial Revolution came about, this was in England, the pollution supposedly caused the lichens on the trees to die, and so the trees were now darker because the bark was exposed. And so suddenly, being a darker moth would blend better with the tree than being a lighter moth. So the light moths did not turn dark, but suddenly the darker moths survived better, had more offspring, and so genetically the population shifted towards one extreme, being darker. And so that's an example of that one. Stabilizing selection is where average is best. Here's one here showing a trait of having either small or large size in these flies, but the wasps can eat the small ones and the woodpeckers eat the big ones, and so what ends up happening is the average is best, and we see that. Another really good example of this that we see is the correlation between sickle cell and malaria, and we talked about this in genetics. So the idea is that having sickle cell is a recessive gene, and if you're little b, and particularly in an area of Africa where, let's say, there's not a lot of medical care, your chances of survival are very low. You may die before you're 18, you may not have any children, and so little b is not going to get passed on. However, we also know that malaria is very common in Africa, and malaria is not genetic, it's carried by mosquitoes. But for whatever reason, if you catch malaria, it can be deadly. So people who are big B are more likely to die if they catch malaria. What we discovered was that people who were big B, little b do not have a lot in the way of symptoms of sickle cell, but at the same time, for whatever reason, this organism that causes malaria does not survive in them as well. So what ends up happening is having one copy of the gene is actually beneficial. And so we've discovered that this little b doesn't get wiped out and actually stays very, very, very common. Lots of heterozygous people in Africa, because it actually gives them some benefit. And so the gene is maintained rather than being wiped out, even though in a homozygous condition it's a negative trait to have. And that's the map here showing how common sickle cell gene is here and how common malaria is here. Okay, the next one, disruptive selection. This is basically where both extremes are favored and average is the worst. For example, here they're showing light and dark coats in mice, and having a medium coat is the worst. And so over time, we end up with a population of light mice, a population of darker mice, and nothing in between. Another example of this here on the right is showing butterflies and how we have natural selection occurring, and so over time, the medium colored butterflies disappear, and we end up with light and dark. A lot of times you're not going to see this in the same area because it's not going to make a lot of sense, because what's going to happen is, if you think about it, aren't you still going to have these guys and these guys mating with each other, which would maintain these intermediate forms? So a lot of times what you'll see is this occurring over, for example, a geographical region, where you're going from north to south, and in the north, let's just say this one is favored, and in the south, this is more favored. And over time, we see these get sort of wiped out in both regions, so that eventually they behave as two separate species. But we see that a lot. It's typically when they're isolated from each other, because then their gene pools are not mixing, and sometimes even if you put them back together again, they'll no longer mate. So that's disruptive selection. All right. Ways we study evolution. I'm going to run through this really, really quickly. Fossils would be one way we study evolution, obviously looking at really old stuff, bones, whatever. We don't see living dinosaurs, but we can learn a lot about dinosaurs, where they lived, etc., by looking at fossils. Paleontology is just the fancy name for the study of fossils, and biogeography is the study of the geographical distribution of species, which would include also the geographical distribution of different fossils that we've found. This is just some pictures of different types of fossils. And then a little quick thing about radioactive dating. Isotopes, remember, all elements are isotopes. It's not like there's an isotope of carbon and then there's normal carbon. Every carbon has 3 isotopes. Hydrogen has 3 isotopes. Most Most elements have at least 2 isotopes. What's unique about a radioactive isotope or a radioisotope is that it all has to do with extra numbers of neutrons or different numbers of neutrons, and in radioactive ones they have enough extra neutrons that it makes their nucleus unstable, and so they break down. They break down at a rate that we're aware of called a half-life, and this differs for each different radioactive isotope. And so basically you have a certain proportion in the atmosphere and it's breaking down over time. For carbon-14, for example, you're alive, you're eating, you're a carbon-based organism. So you're taking carbon into your system. You're eating carbohydrates, proteins, lipids, all those organic compounds, those are being incorporated into you. And so you're incorporating carbon-14 into your body. At the point that you die, you're no longer incorporating new carbon-14 into your body, and what's there is decaying. In the case of carbon-14, it goes through a decay that it goes from carbon-14 to nitrogen-14, and its decay rate is give or take 6,000 years. That's the half-life. And so over time, what you're going to see is something that looks like this on the left. If you were to start with, let's say, 100 grams of carbon-14, and no nitrogen-14, let's just say, in this particular organism, what's going to happen over time is when one half-life passes, the number of grams of carbon-14 is going to be cut in half, from 10 to 5, and the rest became nitrogen. Then that'll get cut in half again, and then in half again. This particular example is actually strontium-90. So the half-life of strontium-90 is 29 years, and so what we would see is this would be 29 years, another half-life, now we're up to 29 plus 29, then 29 plus 29 plus 29, etc. Eventually you get to such a small amount that we can't really use it anymore because it's not enough for our equipment to accurately measure. And so that's why you wouldn't necessarily use, for example, strontium-90 would not be a very good isotope to use for dating very old fossils. Carbon-14 is not great to use for really old fossils. I think it's only accurate up to about 40,000 years, and beyond that there's not really enough. But if you notice some of these other ones, for example, uranium-235, 710 million years, you could certainly date something like dinosaur bones when something decays that slowly. So keep in mind what a half-life is. Just understand that it's not that half is gone and then the other half is gone. It gets cut in half each time. So if 3 half-lives have passed, you would say, okay, so that would be 1, 2, 3. If I tell you the half-life's an hour, then it would be 1, 2, 3 hours. If the half-life is a day, it would be 1, 2, 3 days. If the half-life is 6,000 years, it would be 6,000, 12,000, 18,000 years. So whatever the half-life is, just count how many half-lives have passed and then multiply it times whatever the half-life is. This is just showing fossil records. Keep in mind with these, and I've said this in class, that think of this as a puzzle, and you've got 300 pieces and it's a 1,000-piece puzzle. People argue, well, where's the proof that this little thing here became a horse? And the truth is, fossils support it, but there's a lot of fossils missing. So we found some leg bones. We haven't necessarily actually found entire skeletons of this, and then this, and then this. We found bits and pieces, and then we're putting this puzzle together. But this is just one example of something Darwin would have seen, for example, with the tortoises. A trilobite is a common index fossil. It's something that we know the age of. They're very common. You can go to like Stone Mountain in Georgia and actually buy little trilobite fossils for yourself. The idea is if we find an area and there's a lot of trilobites there, it would also tell us the general age of other stuff we find there. And this is Archaeopteryx, which if you look online, you're going to see that this is not the best example. I think there's a lot of disagreement about this, but this is an example that's been used in the past to show reptile and bird being related to each other, because Archaeopteryx had a beak like a bird, feathers like a bird, but it had teeth in its mouth still, and it had claws on its wings, and it actually, supposedly, it didn't fly, it like glided more like a flying squirrel. And so some would say, hey, is this some kind of missing link between reptiles and birds, showing that birds evolved from reptiles? This is showing geographic distribution. There's fossils that indicate all 3 of these came from the same ancestor, but they were separated from each other. South America, Australia, Africa, you're going to see different climates, different selective pressures, and so over time these would have developed through isolation, mutation, etc., into 3 separate organisms.